Bum, 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 bum. Snap. Alright, we got a 24-year-old female.
That's a male.
I think he's dead. Alright, Watson. What kind of evidence do we have so far? We have victim's blood, right?
Yeah.
Wait. I think I found something. I think it's male gamete.
Crick, that's conditioner.
Watson, what do you got there?
I think I got the hair of the killer.
I like big butts and I cannot lie. Other brothers can't deny.
The hardest part of the job. Hello? Yeah, you want Alan? Yeah, he's kind of busy right now. Yeah, he'll call you back later. It's awkward.
Evening, detectives. How's it going?
Oh wow. Doctor.
How's it going, detective?
What's going on?
Welcome to my home office, guys. Sorry for the mess. I just came back from the lab. Big, busy day there.
It's okay.
Have you guys determined who the killer is?
That we have. Let us debrief you guys.
So basically what we're trying to do is match the DNA found at the crime scene with the DNA of one of the suspects using gel electrophoresis.
We hypothesized that if the suspect's DNA RFLPs are run through the gel electrophoresis, none of the bands that most closely match to those from the crime scene, determined by RF value analysis, must determine the identity of the murderer.
We first took a sample of each suspect's DNA. That was easily obtained through the hair, blood, and other bodily fluids that you guys gave us. Now imagine this. Human DNA consists of over 3 billion nucleotides, where more than 99% are identical among all people. However, it's that one measly percent of code variation that makes us all unique. That 1% therefore gives us our own DNA fingerprint. These variations are called polymorphisms. When DNA restriction enzymes cut at these polymorphisms, we received DNA fragments of different lengths because the enzymes cut at specific nucleotide sequences. We quantify the difference in fragment lengths to create a DNA fingerprint and call these resulting pieces restriction fragment length polymorphisms, or RFLPs, or RIFLIPs. So how do we use these RIFLIPs, you ask? Well, after the restriction enzyme cuts the DNA, we can sort the RFLPs using gel electrophoresis. This method separates the fragments on the basis of size. We first mix the DNA in a buffer solution that makes the resulting solution more conducive to movement in a charged gel. On the gel plate, we have wells where we can place the cut-up DNA. That cut-up DNA was placed on the negative end of the plate. Once the charge ran, the negative charge of the DNA's phosphate backbone was repelled and attracted to the opposite, positive end of the gel plate. The pores of the agarose-based gel filtered the DNA running through the gel. Since smaller RFLPs can wind through the pores more easily, those groups of shorter fragments were found closer to the positive end of the gel, while the larger fragments moved less. As a result, we came up with our own DNA fingerprint, a barcode, as some might say, that showed more of the larger RFLPs on one end and more of the smaller RFLPs on the other. Since DNA sequences are unique to every individual, the crime scene's DNA pattern on the gel should match very closely, but not exactly, to the murderer's DNA. The match cannot be exact because of procedural variation, but the statistical probability of the match, where the two samples match accurately within 1 in 10 billion people, effectively determined who done it.
Okay, cool.
Alright, so this is what we did. The experiment is split into two parts. The first part, you label the 5 microfluid tubes, crime scene, suspect 1, suspect 2, suspect 3, and suspect 4. Then using a new micropipette each time, you add the following reagents, DNA, buffer, restriction enzyme, in respective order and amounts, making sure that the DNA added matches the label on the tube. After all reagents are in place, insert all tubes into a microcentrifuge and spin for approximately 30 seconds. Extract and incubate all tubes at 37 degrees Celsius for 1 hour. After 1 hour, add 2 microliters of the loading dye to each tube. Incubate all 5 tubes at 65 degrees Celsius for 5 minutes in order to stop enzyme activity in tubes. Part 2. Place the e-gel tray inside the electrophoresis chamber and perform a pre-run for 2 minutes. Once the pre-run is complete, load each DNA sample into a corresponding lane. Make sure not to pierce the bottom of the wells with the micropipette tip, and make sure not to overload the wells. Run the DNA for 30 minutes on clone well agarose 0.08% setting. After 30 minutes, stop the electrophoresis run. With the UV cover on, turn on the UV light and mark the DNA bands on the gel.
Here's the data.